guys, and welcome to IEV Online. Javed Khan Afridi here, and you are watching AS Level Biology, and we are discussing the topic cell cycle. This is lecture 5. And now we will start from the end of mitosis, which is telophase, the last stage of mitosis. And after telophase, we will discuss about the cytokinesis, which is the last step of cell cycle. Mitosis, I told you, is a type of nuclear division. Once the nucleus has been divided completely at the end of telophase, which we are studying right now, then the process of cytokinesis will be studied, which is the division of cytoplasm. So what's going on in telophase? Obviously, you know that telophase is the reverse of prophase. Prophase, which was the first step of mitosis, and the different events which we have studied in prophase will be reversed now in telophase, because we have to form two nuclei, and each nucleus have now the original number of cell contents, the DNA contents. And then cytokinesis will also be taking place to divide the cell, each having a single nucleus with the same genetic material. So what's going on in telophase? Let's talk about in some detail about the telophase. Now in telophase, most of the steps of the prophase have been reversed. Like, at the end of prophase, what we have discussed, let me to share once again these topic which we have discussed in the prophase. I told you that in prophase, that this is the largest step of mitosis, but the last step which we have discussed was the formation of spindle fibers called the mTOQ. And I also told that the formation of spindle fiber formed from the tubulin protein, which is a kind of globular protein, and then from the alpha tubulin and beta tubulin. So we say that at the end of prophase, the spindle fiber have been developed, although the spindle fibers did not complete its final shape, but they were in the ester form, and they have been originated from one pole to the other pole, and they start growing toward the centromere of the nucleus. So whenever you want to revert the cycle from the end point to the beginning, because the reverse means the reversion means to rotate the cycle in the backward form, in the backward direction. So the last step is the spindle formation. So what will we see in telophase? Dismantling of the spindle fiber. So first of all, the spindle fiber will be disappeared. They will be dismantled. And then, if see the other steps, like the two centromeres are migrated to the opposite poles. So centromeres, of course, they are still present, and each centromere have connected with the chromatids having spindle fiber. So since the spindle fiber have been dissolved, but the centromeres are still present. So we said that two centrosomes are present, and I say that two mTOQ are present in the cell at each pole. It says that the disappearance of the nucleolus. So now the nucleolus will be reform again. In each nucleus we have the nucleolus. Nuclear membrane is fragmented, so a nuclear membrane will be also reformed. We say now that the two DNA have been migrated to the opposite pole, as you have seen in this diagram. As I told you that the DNA have been reached here at the two poles. This is the last step of, I think, in the anaphase. So what's going on in telophase? That nuclear membrane is formed around each set of chromosomes, sets of DNA. So two chromosomes, two nucleus are formed. The spindle fibers are dissolved. The centromeres still are present at the two opposite poles, and the nuclear membrane also formed. So we say that in one cell now, like in one cell, we have two nuclei to be formed, each have the same genetic material. That's the case which we are studying now in telophase. So during telophase, many of the events of prophase seems to be thrown into reverse. The spindle apparatus is dismantled. Once again, keep in mind here it is the first step of telophase, but it was the last step of the prophase. So prophase is moving back. So when you are moving back, you have to choose the last step from the back cycle. The cycle is ended, now reversal. So when you are moving in reversion, so the last step will be considered as the first step in the opposite way. Now, a nuclear envelope forms around each daughter nucleus. Once the spindle fiber has been dismantled, the DNA is still present, so each DNA will be surrounded by a nuclear membrane. Nuclei appear in each daughter nucleus. Keep in mind, nucleoli is the plural of nucleolus. I mean, we have two nucleoli, because we have now two sets of DNA, and each set of DNA is surrounded by the nucleus. So we say that we have two nuclei now. Now, in each nucleus, there should be one of the nucleolus. That's why I say that nuclei appear in each nucleus. The chromosomes finally steadily elongates. So elongates means they now become uncondensed. As initially in prophase, I told you that each sets of DNA is going to be condensed. And once they're condensed, they are known as the chromosomes. And now here it says that the chromosomes become uncondensed. The DNA become uncondensed, loosely packed, which means it is again converted into chromatin. Chromatin is the uncondensed form of DNA, while chromosome is the condensed form of DNA. So it says elongate as chromatin extend itself from its condensed state until there is once again just a diffuse mass of chromatin fiber. So that's very amazing. Let me to explain some more. Like we have one of the cell initially, this is one of the cell which is a stem cell, and it undergoes the process of cell division. So if it is like moving in cell division, what will be going on? Let me to show some DNA molecule by this red line, and these are the DNA molecule in the chromatin form. So how many DNA here we have? 2 DNA we have. It means this cell have a diploid number of 2. So diploid mean 2 sets of chromosome, 1 chromosomes from father and 1 chromosome from mother. So it has 2 DNA molecule. How many DNA here we have? We have 2 DNA molecule. And how many chromatin? Of course, why we say chromatin? Because it is uncondensed, so we have 2 chromatin as well. Similarly, 2 chromatins and 2 DNA molecule, if this is going to divide, so it must go under the process of interphase, which is since we are talking about the nucleus only, therefore I'm talking about the S phase only, because S phase is involved in the semi-conservative replication of DNA. So here, this is one of the chromatin. Now, what is the actual structure of this chromatin? Like this is the chromatin inside here. So this chromatin inside, if you see it in much more detail, so it is made up of 2 things. One is DNA and one is histone protein. Histone protein. Now the DNA, is not a single structure. It is like, it is like a double helical structure. So it is like this structure originally in the form of like this one in the DNA, but they also have histone protein inside. This is actually the real structure of chromatin having the DNA. Now this is the combination of DNA and histone protein, because we have the histone, here we have the histone, and the red part showing the DNA. So this is the chromatin. So here we have how many DNA molecule? That's why I'm saying 2 DNA, because one DNA is in one chromatin and the other DNA is in the other chromatin. But you know that each DNA made up of two nucleotide. So we say that how many nucleotide here we have. If somebody asks you how many nucleotide we have, I mean how many polynucleotides, sorry, how many DNA strand, or you can say DNA strands. How many DNA strands of our polynucleotide? Here we have 4. Right. Now, what's going on at the interphase step? Each of the DNA should be duplicated, so its exact copy will be formed here. And similarly, the other one as well. So its copy should also be double. So now in this case, we have how many DNA we have? Here we have DNA for chromatin, also 4, and DNA strands we have 8. No centromere until, but because when centromere is formed, then it is said to be chromosome. Now in the next step, which is the prophase. Now in prophase, the nucleus looks like in different phases. The DNA has so become condensed now. Let me just show the other one with the blue one. It looks like this one. It means now it is in the condensed form. So here again we have DNA still 4, but we have now chromosome because it's condensed now. So we have how many chromosomes? We have 2 chromosomes, and we have DNA strands again 8. So DNA strands are 8. And here one chromosome contains 2 DNA because it's now in the double physical form. Now here one thing more, chromatids. How many chromatids now? Now each DNA molecule is called chromatids. We have 4 DNA, so we have 4 chromatids. These chromatids, one of the chromatids is the blue one is the red one, the other one, and these 4 are the sister chromatids. They are located on the DNA molecule. Now, if further we are moving, now this is the prophase. Here is the prophase. But there are so many steps in prophase, like the nucleus may be disintegrated, the nucleolus will be disappear, and so on. And finally, this DNA has been disintegrated. So now, in the next step, these chromosomes will be aligned at the equator, like in this form. Or you can say like it should be horizontally, like let me to show it like in this form. This is one of the chromosomes, this is the other one, and now they are aligned. So here we say that these are the poles, these are the pole, these are the pole, and this is the equator. This is the equator, because equator is showing like they are aligned at the equator, and this is metaphase actually. So they align at the equator, and these are the poles, and on the poles we have here the centrosomes or microtubule organizing center. Then of course this centriole to becomes a bigger one, and they just make the spindle fiber. You can see there is no nuclear membrane because the nuclear membrane has been disintegrated. And then in the next step now, the anaphase. This is the metaphase, where the chromosomes are attached to the spindle fiber but not yet separated. And finally, in the anaphase, the chromosome will be separated from one another. Now this is the let me just draw it some more clearly. So here we have the centrosome on one pole. Here we have the spindle fiber. And here we have the one of the chromosomes to be attached here. Similarly, this is the spindle fiber, and here we have the other one to be attached here. And that's why on the other pole, so we have here, the red one should be here, and then the blue one here. And they have separated the DNA. Now this is taking place in anaphase, and then in anaphase, further shortening of these microtubules start. they are reaching to their respective pole. And finally, I told you in the telophase that the spindle fiber will be disintegrated. And as a result, what's going on? Let me to copy it from here. Now, what's going on in the next step, which is the telophase? This is the pole, the centrosome. This is another centrosome, two centrosomes. The spindle fiber has been dismantled. Here we have one of the chromosome, and this is the other chromosome. Similarly, here we have one of the chromosome. Here's the other one. And now I told you that the nuclear membrane is going to be formed, but the centrosome is outside, because it is not inside the nucleus. So this is one of the nucleus. This is another nucleus. This is taking place in telophase. So yeah, now you are in telophase because you are in just one cell, but there are two nuclei. And now in the next step, what's going on, that these nuclei will become, they have the uncondensed DNA now. The DNA again become uncondensed like from the original cell. And similarly, this other one is also like the uncondensed form. Similarly, in the other nucleus as well. So we have the condensed DNA, uncondensed DNA, sorry, and then the other DNA as well. And the centrosomes, of course, they are also present here. This is taking place in only one cell. So one cell now have two nuclei. Now this is the case of telophase. And now we will start about the cytokinesis. In cytokinesis, what's going on, that the plasma membrane is going to be pinched off from both the sides, like this way. And now this pinching is going to deeper and deeper. Like I'm just going to copy this one. So here now this is again going to deeper and deeper. The membrane is going to deeper and deeper. And the result is like breaking of the nucleus, breaking of the cell in just inside the middle. Again here. And finally, it goes to be joined together and the cell will be divided into two. So two cells will be formed from the one. And this is actually cytokinesis. In this step, we have cytokinesis because now we have two cells instead of one. So in telophase, I told you that we have one cell with two nuclei with uncondensed DNA, and the centrosomes are also present in two, one on the opposite pole. The organelles also become doubled. So many organelles in the cell size is bigger one. And once cytokinesis takes place, all the contents will be halved and two identical cells are produced of the smaller size with uncondensed DNA exactly in the same way like the original DNA. This is what's going on in the cytokinesis. So let's talk about once again about the telophase. So the spindle apparatus is dismantled, a nuclear envelope forms around each daughter nucleus, and then nuclei appear in each daughter nucleus. The chromosome elongates as chromatin extends itself from its condensed state until there's once again just a prefuse mass of chromatin fiber. At the end of telophase, the cell contains two fully formed daughter nuclei. At the end of telophase, we have two nuclei in one cell. They are genetically identical because they have been formed from semi-conservative replication of DNA, which are the exact copy is formed, from which they were formed. Telophase mark the end of mitosis. This is actually the end of mitosis. The original nucleus has divided into two genetically identical daughter nuclei. And now, next, the cell must divide into two. So in the next step, it is the cell which is divided, not the nucleus, because the cell is also now bigger in size. Not only they have two nuclei, but they also have double number of mitochondria, double number of Golgi body, double number of ER, double contents of cytoplasm. So it's a bigger cell, and now it's the time to distribute the cytoplasm also into two equal cells. That's why cytokinesis, it normally starts in the middle of the anaphase and remain continued till at the end of telophase. but usually it's it's prominent at the end of telophase. So it is simultaneous process with mitotic cell division and not a separate process. During cytokinesis, the original cell divide into two. Now, what's going on in animal cell? This involves the plasma membrane forming a constriction. What do you mean by constriction? This is called constriction. This is called constriction. The invagination of the plasma membrane inside, or the projection of the plasma membrane inward, or the pinching of the plasma membrane is called pinching or constriction. This constriction is because of the forming a constriction across the center of the cell. This constriction is because of the protein filaments. Actin filaments are responsible to make the constriction, which become tightened, pinching the membrane inside in the center of the cell, making furrow. Furrow means inside projection, and finally pinching of the cytoplasm into two cells. So this is the way which I have explained cytokinesis. This is only in case of plant cell. What's going on in the in the animal cell. Now, what's going in the plant cell? How a plant cell undergo the process of cytokinesis? In animal cell, I told you that it's a simple process of the pinching of the plasma membrane due to the actin filaments, and the plasma membrane pinch the cell in the middle, separating the contents of the cytoplasm as well. Now, what's going on in plants? A plant is something, the story is different. Now, in plant, there's no pinching of the plasma membrane. Instead, here is a plate is formed in between the cell. Like if I'm giving you once again the example, here is a plant cell. It has a cell wall. And of course, we have the two nuclei here. They have the DNA in uncondensed form. They also have the centrosome near them. And the organelles are also doubles, the bigger cell now in size. So what's going on? There's no pinching of the plasma membrane. Instead, we have some vesicle formation. Vesicles are formed. And these vesicles are aligned in the middle of the cell, like at the equator region. These vesicles are formed from the Golgi body. These vesicles have carrying specific chemical called pectin. Inside the vesicles, we have a specific chemical that is called pectin. Later on, these vesicles are ruptured and their pectin joined together. The chemicals react with one another to make a plate, a type of plate formation here from the vesicles. And this plate is called pectate wall. So pectate is formed from pectin, and pectin is enclosed in vesicles, and these vesicles are bud off from the Golgi body, which play their role. This is the case in plants, which is the cytokinesis followed by the formation of pectate wall. Now, if we focus again here, cytokinesis in plant cells. So in plant cells, a cell plate forms where the metaphase plate had been. The cell plate is synthesized by the fusion of multiple membrane enclosed vesicles formed by the Golgi body. This fusion supplies new plasma membrane for each of the two daughter cells. So a new plasma membrane along with the cell wall is formed in between the two cells, followed by the plasmodesmata. Synthesis of a new cell wall between the daughter cells then occur at the cell plate. So initially a plasma membrane is formed, but with the passage of time, then a cell plate is formed. Here the diagram you can see. Look at this diagram if you're just going to magnify. So in this case, you can see the DNA is actually uncondensed, not a condensed form. So it's the early stage, it's the interphase. So prior to mitosis, each chromosome makes an exact duplicate of itself. The chromosome then thicken and coil. So here the centriole, this is the nucleus, and there's the duplicated chromosomes. The number of chromosomes have been doubled. The S phase already have been taken place. Then you can see that the cell become condensed now, and once it condensed, you can see the chromosome now instead of chromatin, and each chromosome is the condensed mass of the DNA, because it has the centromere. One centromere is responsible to code one chromosome. So we have here how many DNA we have. We have 4 DNA, but we have 2 chromosomes because we have 4 chromosomes, but we have 8 DNA in this case. 1 DNA here, 1 DNA here. So 1 chromosomes contain 2 DNA, and there are a total of 8 DNA in 4 chromosomes. This is centrosome. This is another centrosome that have been replicated, ester formation taking place in prophase. So this is the early prophase, and now this is the late prophase. What's going in the late prophase? Because the nuclear membrane is going to disintegrate it. The spindle, the centrosomes are reached to the opposite pole. Nuclear membrane disappear, chromosome more condensed. This is the late stage of prophase. And now you can see here is the metaphase, which is going in the metaphase. The DNA or the chromosome have been aligned at the equator. This is equator and this is the pole. So they are aligned on the equator. So that means metaphase. And in the metaphase now, after metaphase, we have anaphase. Spindle formation have been completed. The chromosome have been broken down, and the sister chromatodes are separated and have moved to their respective pole. This is anaphase. And I can see the telophase, where the 2 nuclei have been again reformed. The spindle fiber are going to be dismantled. Pinching of the plasma membrane remain continue. And here we have the cytokinesis, where 2 cells have been formed from 1 cell. That's what's going on in the process of mitosis, and that's the end of cell cycle. Cell division. We have not only dividing, we have not only divided the nucleus, but we also have divided the cytoplasm. So a parent cell is divided into 2 cells. This is what's going on in the mitosis. So the next video, I'll tell you about some important function of mitosis and some more detailed structure of the chromosome and gene and DNA. So stay with us. See you in the next video.